Donaldson, who is our I.B.A.N.G.S Young Investigator Award recipient for 2019. Zoe did her undergraduate studies at UCLA. She got her PhD at Emory with Larry Young, and then did a postdoc with Renee Hen at Columbia. She is currently an assistant professor at University of Colorado Boulder, a fellow Coloradan, since 2016, I believe. And she has joint appointments in two departments there, molecular, cellular, and developmental biology, otherwise known as MCBD, and also in the Department of Psychology and Neuroscience. She has received numerous awards and honors since she was an undergraduate student, including the I.B.A.N.G.S Travel Award, when it was in Boulder, ironically, in 2012, when she was a postdoc. And she also, more recently, has received a couple of very prestigious awards, the NSF EDGE grant, and also the NIH Director's New Innovator Award. These are really important, prestigious awards, and give her a lot of flexibility as well as money to pursue a lot of different things. I'm just going to read something off of her website that kind of describes what she's doing. She's interested in studying the mechanistic basis for individual differences in behavior and how these impact likelihood of developmental illness. Of course, we knew she was doing that. She uses really cutting-edge techniques, a number of cutting-edge techniques such as in vivo calcium imaging and optogenetics. And I also just want to read a couple of titles from some of the public talks that she's given that kind of help describe the kind of work she's doing. One was Lost in Loss, a window into the grieving brain. Another is Decoding Monogamy, the neuroscience of social bonding. And lastly, of modulators and monogamy, molecules of monogamy, sorry. And then a couple things, I told her I was going to tease her with this a little bit. This is on her CV, so I'm allowed to do this. She was a participant in a workshop for science fiction filmmakers, sponsored by the National Academies of Sciences. And also she was involved with something called an improv comedy session with science. So that's a little bit of a tease. It gives you a little bit of insight perhaps to her life outside of science, maybe not. One last thing is that she's working with a model organism, a sort of, I guess you might call it an uncommon model organism, and has been developing a lot of genetic tools to work with this animal, and I think she's going to talk a little bit more about that. So please join me in welcoming and congratulating Dr. Zoe Donaldson.
Thank you so much for that warm welcome. So science and improv, I'm not an improv comedian. Instead, I volunteered to give a talk that wound up being the fodder for improv. So if you really want to see your research go in a bizarre direction, just let a couple of improv comedians start using it for their performances. So I wanted to start today by addressing something that I think is actually very, very common. And that's when the request for nominations for this award went out. I looked at it and said, oh, you know what, I'll ask someone to nominate me next year because I'll have a more impressive CV. I mean, that's just a given, right? Time passes. And fortunately for me, Gene Robinson emailed me and he said, I'm nominating you for this. And it's a good thing he did because here I am standing in front of you today. And so I want to say thank you to Gene and also thank you for the organizing committee for what's been a very inspiring meeting so far. So the sort of big overall framework that underlies my work is sort of, why does behavioral diversity matter? Why is it even worth studying? And I would argue that there's two reasons why it's worth studying. The first is from an evolutionary perspective. So I'm very interested in understanding how changes at the genetic level have contributed to the tremendous behavioral diversity that we see existing within the world around us. But I think it also matters from the perspective of mental health. So we know that there are these ingrained, innate behavioral differences that are in part guided by genetic differences. And it's thought that these contribute to predispositions for mental illness. But that link between genes and behavior with respect to mental illness has grown as we've gained sort of technologies to perform GWAS and other more sophisticated analyses over time. But what we still lack is a good understanding of how these specific genetic variants can act within the brain to change physiology and what this means in different brain regions and across the life of the organism. And so today I'm going to focus on two stories The first one is through the lens of evolution. And what I'm going to show you is that repetitive DNA elements, especially when located in promoter regions, can contribute to differences in gene expression and socio-behavioral diversity across and within species. And this is work that's really inspired by work that was performed initially in voles. I'm then going to switch over, using sort of the same lens of linking genotype to phenotype, to talk about its relevance within the context of mental health, specifically focusing on how development and how different brain populations within the brain can be sensitive to different common genetic variants. Finally, I'm going to finish up with some lessons learned along the way. I think we all know this, but I just will reiterate that it turns out that mice are not tiny humans. And from what you'll see today, it turns out that they're also not tiny voles. All right. So I'm going to talk about two separate neuromodulatory systems over the course of this talk. The first one here is vasopressin. So vasopressin is a highly conserved hormone that's made within the hypothalamus, and it's been implicated broadly in modulating social behavior across a wide array of species, but where it's most famous is for its role in stimulating pair bonding within prairie voles. In the second half of the talk, I'm going to focus on serotonin, another ancient neuromodulatory chemical that acts on diverse receptor populations within the organism to modulate a wide array of different behaviors. And so to start us off talking about vasopressin, I need to give you a little bit of background on these guys. So these are prairie voles. They've become somewhat famous. I think they're in a lot of textbooks at this point. And the reason that they stand out within the rodent or even the mammalian kingdom in general is because these guys are monogamous and biparental. They fall within that 3 to 5% of mammalian species that are capable of forming mating-based pair bonds. And one of the nice things about this system is that we can directly compare them to a sympatric species, the meadow vole, which looks nearly identical but has very, very different social behavior. So these guys are promiscuous and uniparental, so unlike these animals where they'll mate and stay together for life, this one, the female will mate and she'll go off and she'll raise the offspring on her own. Now within the laboratory, we can actually quantify aspects of this behavior by using something called the partner preference test. And so that's what you see here. And so within this test, we have two animals that are tethered on opposite sides of this three-chambered apparatus and a test animal in the middle. And so the first thing that you'll see is in this is a bonded pair. And so the animal goes over to this opposite sex individual it's never met before, gets into a little bit of a tussle. And this is very characteristic of pair-bonded prairie voles, where they really only want to spend time with their partner. And so what you'll see here, as we'll zoom in a second, that the behavior towards their monogamous partner is quite drastically different than what we saw with that opposite sex novel individual. And the nice thing about this test is that it provides us a simple quantitation of this behavior. So we can simply ask, how much time did you spend with your partner versus the novel individual? And if they formed a bond, you'll see that they spend substantially more time with the partner than with the stranger. If they haven't formed a bond, you'll see that they don't differentiate between these two individuals. If you did this test with a mouse or a laboratory rat, you would see basically this behavior here. Now, one of the nice things that we know is that mating actually facilitates this. And this is my undergrad who has to quantify a lot of mating, so he made a GIF, and it's now showing up in everybody's presentations. But this is from an actual paper where literally all they did is show that sex facilitates partner preference formation, and if they don't have sex, they don't form a partner preference. Now, the nice thing about this is it creates two conditions, sort of a gain of function and a loss of function condition. And so what we can do is we can create conditions where they mate, so they should form a bond, or they don't mate, so they don't form a bond, and then we can ask about the necessity and sufficiency of different molecules for this behavior. And so in work that was done in the sort of mid-90s, we know that you can infuse either oxytocin or vasopressin into the brains of these animals, and even in the absence of mating, they'll still form a partner preference, suggesting that this is inductive for this process. And likewise, you can create conditions that are normally conducive to partner preference formation, but you can block the signaling of either oxytocin or vasopressin, and it will block the formation of a partner preference. Now, one of the things that I find puzzling is that this only works in prairie voles. If you infuse oxytocin or vasopressin into the brain of our promiscuous species, the meadow voles, all they do is groom themselves a bit more. So the question is, why do you have such profoundly different species-level effects of this hormone on behavior? And it turns out that it's not the hormone itself, which is actually incredibly highly conserved and produced by the same neurons in the brains of animals ranging from fish through mammals, that it instead has to do with the distribution of the receptors within the brain. And so if we look at these two animals here, what you can see in an autoradiographic image is that we have higher levels vasopressin receptor within this area of the brain called the ventral pallidum. And I'm going to show you a number of these types of images today. Basically, it's using a radioactive ligand that binds to the receptor, then this is exposed to film, and the take-home message is the darker the region, the higher the level of the receptor. Now, one of the really interesting things is that we know that this difference in receptor level between these two species is actually responsible for driving species differences in partner preference. We know this because if you put an antagonist into this brain region, you can block partner preference in our monogamous species, but in somewhat more exciting studies that got a tremendous amount of popular press, you can increase the level of vasopressin receptor within the ventral pallidum of these guys, and they'll form a partner preference, indicating that the circuitry exists and it's all about how you use it. So what I was interested in is identifying the genetic elements that were responsible for driving these species differences in receptor patterns within the brain. And so this was a point in time when we didn't have genomes, we had extremely limited resources, but one thing that we could do is we could fish the gene out of a library. And when we did this, we found that the coding sequence was nearly identical across these two species, but that when you look upstream of the gene itself, there's this repeat-containing element that's hundreds of base pairs longer in prairie voles than it is in meadow voles. And somewhat intriguingly, but not surprisingly, this repeat-containing element is also variable amongst prairie voles. So you have a spectrum of different lengths. I'm showing you here just two of them. And this variation in this repeat-containing element is actually associated with individual behavioral differences across prairie voles within the lab. And so as a graduate student, I became interested in the hypothesis that maybe this repeat element was directly driving these differences in gene expression patterns in the brains of these two species and potentially also contributing to individual differences in receptor patterns. So obviously we CRISPR'd it, but we didn't because there was no CRISPR at the time. This is just an opportunity for me to show you that I made the first transgenic prairie voles using lentivirus as they glowed green. It was fabulous, but this was not a technology that we could have used at the time to approach this question. So instead what I did is I turned to another organism that was more genetically tractable. In case you're having any trouble following, this is a prairie vole, this is a mouse. And so at the time we had the ability to go in and specifically genetically engineer the genome of mice, making very precise edits. And so what I did is I generated a series of mice in which I replaced the upstream region of the mouse vasopressin receptor 1A gene with corresponding sequence derived from prairie voles. I then generated 3 separate lines that differed only in the content of this repeat-containing element. The locus was otherwise genetically identical, and this was important because it allowed me to assess the direct effects of this repeat-containing element independent of any potentially linked variants. So the first question that I had after spending years and years doing southern blots is, what do these mice actually look like? And so here I'm showing you 2 levels of the brain. So this is in a prairie vole, this is in a wild-type mouse, this is just a bit farther back. And you see that between voles and mice, there are again striking species differences in receptor patterns. And this is what my mice with the prairie vole promoter region look like. Now, I won't lie, there might have been a little bit of crying in the dark room when I developed those films, because this definitely looks more like a mouse than it does like a vole. But I took a deep breath, and what you can clearly see here is that there are some brain regions in which vasopressin receptor patterns have actually been altered in these mice containing the prairie vole promoter. So we can see an elevated level of the receptor within the dentate gyrus, the thalamus, and the central amygdala. And this was a blessing in disguise, because what this allowed me to do was subsequently focus on these 3 brain regions to ask whether or not the repeat element was responsible for driving differences in receptors specifically within these brain regions. And what we know from this is that if you want to explain the full pattern of vasopressin receptor in voles, it has to be due to elements that are not only present within this proximal region, but also elements that are more distal within the genome. And this is probably something that seems very obvious at this point, but at the time that we were doing the work, I don't think that was as well accepted. So the first question I asked was just whether or not species differences in this repeat-containing element could drive differences in receptor patterns within these 3 brain regions. And so here I'm just giving you reference images from the prairie vole and from the meadow vole, encompassing those 3 brain regions that I mentioned. I then compared mice that were homozygous either to the prairie vole or the meadow vole version of this repeat-containing element. And so when I did that, what I saw is that these differences in receptor patterns within these 3 brain regions are indeed being driven by differences in this repeat-containing element. You can see that it's much lower in these 3 brain regions in the mouse line carrying the metabole version of the repeat-containing element. And that the direction of this effect is actually the same as the direction that we see occurring naturally between prairie voles and meadow voles. So then I went on to ask the question of whether or not allelic differences in this repeat-containing element might also contribute to individual variation in vasopressin receptor patterns. Now this was a somewhat more risky idea because the difference between this repeat-containing element in meadow voles and prairie voles is on the order of hundreds of base pairs. And here we're talking about a difference in the order of tens of base pairs. But what we found was a quantitative difference in the amount of vasopressin receptor present within the dentate gyrus. So a much more subtle phenotype, but that fits with the more subtle genetic manipulation and genetic difference between these lines. And so if we put all of this together, what we come to is that there are brain regions where replacing this upstream region and putting in different repeat-containing elements has no effect on vasopressin receptor expression within the brain. There are other regions where changing out this promoter region recapitulates species differences between here the meadow vole and the prairie vole, and in at least one brain region we can also recapitulate individual differences in expression patterns. So I think I'm not going to harp on this. There were some subtle behavioral differences between these animals, but those behavioral differences were related to stress coping and anxiety, which is what you might expect from the regions where we saw differences in receptor patterns and not related to differences in social behavior. So together, I think what this has taught us is that inter- and intra-species differences in the V1A promoter drive receptor patterns in specific brain regions. And so this is starting to build a framework in which we can think about gene expression patterning within the brain as this sort of combinatorial code in which you're using different enhancers in different brain regions. And as such, a polymorphism can have an effect in one or a subset of brain regions without affecting expression in other brain regions. And the other thing is a more sort of general lesson learned is that these repeat elements are highly mutable. They change rapidly over evolutionary time because of slippage of the DNA polymerase. And so they expand and they contract, and when they're located within regulatory regions of the genome, they actually represent an evolutionary mechanism for generating diversity in gene expression that can then be acted upon by natural selection to shape behaviors within a population. The one thing we did not learn, that I wanted to learn when we started, is what are the genetic elements that are actually responsible for driving the species difference within the ventral pallidum, which we know is so important for the formation of partner preference. And I'm happy that sort of genomic technologies have come so far along because I think it opens up the possibility that we can cast a much broader net to potentially try to identify enhancers and other elements that differ between these species that could explain this species difference in receptors. Now, as I was sort of carrying out this work, I became very interested in the idea of how this might actually relate to us as humans. And so when I was looking for postdoc experiences, I decided to join a lab where I could sort of switch from looking at genotype to phenotype through the lens of evolution to now asking questions of how genotype and phenotype are relevant for human mental health. And for this, I decided to focus on another ancient system, the serotonergic system. And the serotonin system is different from the vasopressin system in a number of ways. Well, one of the big differences is while vasopressin has a few receptors that can vary tremendously in their spatial patterning in the brain across species, serotonin has taken a different route, where instead of having a lot of variation in receptor patterns, what we see is variation in the number of receptors that serotonin can actually act on. So within humans, there are 14 known receptor subtypes that serotonin can act on. And I chose to focus on one of these, the serotonin 1A receptor, because it's been extensively implicated in mood and anxiety. If you knock this gene out, you make anxious mice. So one of the things that's been tricky about understanding the role of serotonin 1A receptors diversely in mood and anxiety has been the fact that it's actually really widely expressed within the mammalian brain. And within the mammalian brain, it comes in two general flavors. So we have autoreceptors. These are actually serotonin 1A receptors that are located on the serotonergic neurons themselves. And these neurons act as a brake within the system. So when serotonin activates these inhibitory receptors, you get a decrease in the amount of serotonin that's being released widely throughout other brain sites. In addition, we have heteroreceptors, and so these are widely expressed throughout limbic brain regions. And so these are downstream receptors that can mediate the effects of serotonin on diverse behaviors. And so what I wanted was a system that would allow me to independently modulate the level of these different receptors, autoreceptors versus heteroreceptors. And so I turned to some of the fancy transgenic tools that existed in mice, still exist in mice. I decided to use a system that consists of 2 transgenes. So the first transgene is just knocking a TETO site upstream of the endogenous mouse HTR1A gene. This doesn't in and of itself do anything. These mice have normal wild-type patterns of serotonin 1A receptor levels. But the power of this system is that it can now be combined with various other systems in which we can use a cell-type specific promoter to drive a tetracycline transsuppressor. And when we do this, you can express TTS within this cell type within the brain. It lands on the TETO and it effectively shuts down serotonin 1A receptor expression, specifically within these cell types. And the nice thing here is that this is not ectopic, so we're just damping down expression in specific subsets of cells within the brain from the endogenous gene itself. Now, the nice thing about this system is it not only provides cellular specificity through the use of different promoters, but it also provides temporal specificity because at any time you can add doxycycline to the system, and when you do that, doxycycline sequesters TTS such that it no longer can bind the TETO and suppress expression. So I'll just very quickly show you an example of what this can do. We can use a forebrain specific promoter for our TTS, which effectively downregulates or suppresses serotonin 1A receptors within the forebrain. And again, these are just autoradiograms, but with a ligand for serotonin 1A receptors. But it has no effect within the midbrain. If we use here a serotonin neuron-specific promoter, the forebrain populations are left intact, but you see a reduction within the RAF-A. At any given point in time, we can actually add doxycycline into the system, which we do by putting it into the animal's food, and you can see that we can recover wild-type levels within the forebrain or the midbrain. So this is a really nice system for asking about pleiotropy within the brain because it allows us to suppress different receptor populations. And so what we know, or what we knew sort of when I came into this, is that if you knock down the receptors within the RAF-A, you get a mouse that's more anxious, but it has no effect on depression-related behaviors such as stress coping. Conversely, if you knock down the heteroreceptors within the forebrain, it has no effect on anxiety, but has a series of effects that are seen as pro-depressive, so more passive stress coping. And so there's this nice dissociation between the role of the autoreceptors and the heteroreceptors on different aspects of mood and anxiety. But this has been knocked out throughout the animal's life. And one question I had is just, is this an ongoing sort of necessity that you have autoreceptors, or are they acting within a developmental sensitive window? And the reason that I was interested in this question is because there had been a pharmacological study where they had applied antagonists postnatally around this time frame, from postnatal day 10 to 30, and they found that these mice, where they blocked signaling at serotonin 1A receptors, had increased anxiety levels, but that the application of the same antagonist in adulthood had no effect on anxiety, suggesting that there might be a sensitive window during which serotonin 1A receptor signaling set up anxiety levels throughout life. And I wanted to know whether or not this could be due to the autoreceptors. And so, taking advantage of both the temporal and spatial gene control that was enabled by my transgenic system, what I did is I essentially suppressed autoreceptor levels just around that same window where the pharmacology was applied. So to give you a sense of what that looks like, on the left here are controls, and on the right are our knockdown animals. And you can see that at postnatal 14, these guys are the same. But by day 21, we have about a 40% reduction in serotonin 1A receptors within the RAF-A, but that this is fully rescued by postnatal day 30. So the point that I want to make here is that this is something that is very subtle in magnitude. We have only about a 40% decrease in receptor levels, and it's brief in effect. And so I think it was a little bit surprising to me then that these animals are also more anxious. And so this was seen in a variety of tests. These animals with postnatal knockdown spend less time and travel less on the open arms of the elevated plasmase, and likewise they spend less time and they travel less in the anxiety-provoking portions of the open field. So what this allowed me to do was sort of refine our understanding of these systems such that we now know that autoreceptor knockdown or knockout increases anxiety only if it occurs during development. My colleague did the corresponding study in adulthood, and what she found is that there's no effects on anxiety in adulthood, but that she actually gets sort of a resistance to a pro-depressive phenotype. So if we put all of this together, what I think it tells us is that there are really important considerations, unsurprisingly, for where in the brain things are happening. So a single gene can have very different effects in different brain regions, contributing to its pleiotropy. But now we also have to think about this through the lens of development, where we can have very different effects from the same manipulation at two different points in time. So I became fascinated not only by the idea that where and when matters, but also that these were very subtle changes in receptor levels, 20 to 40%. And yet they were behaviorally meaningful. These mice were more anxious. And when we start thinking about sort of the context of psychiatric and mental illnesses, we're not talking about people walking around with gene knockouts. We're talking about wanting to be able to understand how naturally occurring variation within human populations might be contributing to individual differences in behavior. And so what we know from the studies that I've shown you thus far is that the small changes in receptor levels can affect behavior with important spatial and temporal specificity. But I sort of wanted to know what might this all mean within the context of psychiatric illness. So one thing that I think is really nice is that we know that there's tremendous variation in serotonin 1A receptor levels within humans. We know this from PET images. This is just a PET study sort of showing you two extremes, someone with high levels of serotonin 1A receptor and low levels. You see the same thing if you look at the mRNA or the protein levels in post-mortem tissue samples. So this is a system where this variation already exists, and so in mice we know that this variation is meaningful, and so it's a perfect system for asking what are the mechanisms that contribute to these individual differences in receptor levels. And so sort of halfway through my postdoc, I transitioned to focusing on one potential driver of individual differences in serotonin 1A receptor levels, and that's a SNP, RS6295. So this SNP, you can tell from the low SNP number, has been around for a while. It's well studied, and indeed there's a meta-analysis that suggests that this SNP is associated with depression risk, that the G allele at least is associated with depression risk and increased suicide attempts. And I was able to replicate that in a primarily African-American sample from the Grady Trauma Project. So here we have a sample of about 1,300 individuals, and we see that individuals who are homozygous for the G allele report, a greater percentage of them report attempting suicide and being hospitalized for psychiatric reasons. And these two are intertwined because the primary reason for psychiatric hospitalization is suicide attempt. And so this was a nice verification of what had previously been primarily seen within Caucasian populations. And these odds ratios, if you're familiar with association studies, are actually surprisingly high. So the other reason that I was interested in this SNP is that I think it's not just a marker. And the reason that I say that is because this SNP is known to alter the binding of a transcription factor neuter DEF1. And so this is work that had been done by my collaborator, Paul Albert. Here in a biochemical assay, you can see that the C allele is effectively competed off by application of neuter DEF1, but that the G allele is unaffected. So this means that this may actually be a functionally relevant SNP. So I started thinking about this within the context of the brain and what we knew about these transcription factors, and it turns out that it's not a simple story of up or down. So neuter DEF1 has different activities in different brain regions. So within the raphe, neuter acts as a repressor. So if it can bind the C allele, it's going to reduce levels of the receptor within the autoreceptor population. Conversely, it actually acts as a transcriptional activator within the forebrain, the result of which is that you would expect it to increase levels in the heteroreceptor population. So in other words, what we would expect from the C allele, our functional allele, is that we have a decrease in the autoreceptors and an increase in the heteroreceptors, and that when we abolish the binding site with DEF1 by the introduction of the G allele, we actually see an inversion of this effect. So we see more mRNA being produced from the G allele in the autoreceptor population and less within the heteroreceptor population. And so this was a model that I felt that I could test. And in order to do so, I took advantage of allele-specific sequencing. So essentially it works like this. You have your genome from a person who's heterozygous for the SNP, and then you measure the amount of mRNA being derived from the G allele compared to the amount being derived from the C allele, and the relative area after the peak here tells you the relative amount of mRNA from each of these. You can do this, so there's two things that are really nice about this. So the first is that these alleles are in the same nucleus. They're in the same transcription factor environment, which means any differences in expression have to be due to differences in cis, such as RS6295. And the other is when working with postmortem human tissue, there are a lot of variables that are outside of your control. One of those is postmortem interval, but there are a variety of others. And so having both of these alleles allows a within-individual comparison that I think is somewhat more robust to the confounds of things like postmortem interval. So use pyrosequencing. I'll just give you some example pyrograms here. So in this pyrogram, you can see that we're detecting about 63% of the mRNA coming from the C allele and 37% from the G allele. Reassuringly, we use the genome as a control where we know that our C and our G allele are present at 50% levels, and we see that our pyrogram is at 50-50. And so just to remind you that now that we have this technology, what I wanted to know was whether or not we observed this relationship, essentially an inversion of the G to C allele ratio in the autoreceptors relative to the heteroreceptors. Now it turns out it's relatively easy to get postmortem human prefrontal cortex from controls, and so this is a relatively large sample size, and reassuringly what we see is across all the individuals in this sample, we're having more mRNA produced from the C allele than from the G allele, as would be expected from our model. We also see that, albeit blunted, within the hippocampus, but it goes away entirely within the midbrain. Now, I'm not entirely sure why we don't see an inversion of this ratio within the midbrain, except to say that there are heteroreceptors within the midbrain, meaning that there are serotonin 1A receptors within the midbrain that are not expressed on the serotonin neurons themselves, and so this may be masked by cellular heterogeneity, or our model could be a little bit wrong. So what we know now, if we put together all of the mouse and human work that I just showed you, is that the where and the when of gene expression actually matter. That individuals who are homozygous for the risk allele have an increased risk for psychiatric hospitalization, and that the G allele is associated with decreased mRNA production in the human prefrontal cortex. So I like that this story works its way through multiple models using multiple techniques and putting together what I think of as convergent evidence. But the thing that I was dissatisfied by is that it's going to be very, very difficult to determine the direct effects of this SNP in either post-mortem human tissue, where you're necessarily limited at certain developmental time points, or in cell culture, where you can't look at things like behavior. And so I decided that the approach that I was going to use looks very much like one I've already shown you, and that's to make mice that actually contain this allele. So here I took a slightly different approach. I thought about making knock-ins, where we essentially replaced the upstream region of the mouse gene with that from humans, but by this point in time we had a much broader understanding that the regulatory network for a gene can be quite spread out throughout the genome. And so I decided to take a different approach. I used a human bacteria artificial chromosome, so about 180 kb of human sequence that contains the human serotonin 1A receptor gene in the middle of it. And what I did is I wound up duplicating that gene such that one copy of it is flanked by LOX-T sites and the G allele is flanked by FRT sites. And again, an important feature of this is that the only difference between these is RS6295. So we don't have to worry about linked variants and their potential effect, or the potential effect, combinatorial effect of a haplotype. So I created founders where I randomly integrated this into the genome, and then I bred those founders successively to a flip animal, which gives us the C allele alone, and then to a Cree animal, which gives us the G allele alone. I then bred these onto a serotonin 1A receptor mouse knockout background, so the only source of serotonin 1A receptor within these animals is from the human allele, and then I brought the alleles back together such that all of the data that I'm going to show you today is animals that were bred from GC heterozygotes. So once again, we're back in the dark room asking the question, what do these mice look like? And so here I'm just giving you some reference. So here's a mouse that's actually heterozygous for the serotonin 1A receptor. I'm showing you the het because, as you'll see in a minute, there's very, very low levels of expression from the humanized mice. And so if I put a wild type up here with the same exposure, it would just be black. And then underneath, this is just to show you that our autoradiography does indeed work. We don't detect any signal when the animals are not making the receptor. And then I had generated 3 independent founder lines. And so there's a few things that are immediately obvious. So one is that none of these lines have the same pattern of expression. All of these lines have much, much lower expression than you see even in the heterozygous mice. And one of these mice is just not even expressing at all. And so I'm not even going to include that mouse because there's no point in studying the knockouts, which have been extensively studied. And I'm instead going to tell you what we did with these 2 lines here. And there is actually some expression here. It's very restricted and very faint in line B. And I think what this tells us is it provides a sort of cautionary tale that where you insert your back, even if it's giant, can have very important effects on your serotonin 1A receptor patterns, or any transgene, presumably. So we've gone to great lengths to make these mice, a lot of breeding, and what I realized is although they don't provide an ideal model for understanding serotonin 1A receptors and looking at things like gene-environment interactions, they're still good for answering one particular question, and that's whether or not RS6295 can indeed modulate receptor levels. And so we started by asking the question of whether or not there were differences in the receptor levels in line A or B. And the short answer is yes, and the longer answer is in only one line. So here you can see that there's no difference in any of these brain regions in our CC or our GG mice in line A, but in line B, we do indeed see differences where the GG animals have higher levels of receptor than the CC animals within these brain regions. And so we see an effect in only one line. So this was a little bit puzzling, but we went ahead and we assessed behavior, and the behavioral differences were incredibly subtle. I'm just going to show you the 2 that reached statistical significance at the p=0.05 level. The first is that animals from line B, the GG animals reared less in the open field, so they reared up less. But there were no differences in line A. And likewise, in the light dark box, the line B, the GG animals traveled less, but there were no differences in line A. So the good news here is that at least we're seeing the behavioral differences in the line that has the receptor differences. But we're still left with this question of how in the world are we only seeing genotype-driven differences in one of 2 lines? And this led me to ask, I've thought about this a lot, but why? And so I think that there's a few potential answers. But I went back to the fact that these guys have very, very different patterns of receptor expression. First of all, these guys have much stronger expression. These guys much more blunted. It's restricted to a couple of brain regions. And the brain regions that are expressing the receptor are largely non-overlapping between these 2 lines. So one very simple explanation might be that this SNP only matters in brain regions where we're expressing the receptor in line B and not in brain regions where we're expressing it in line A. But there are some other confounding factors. So these 2 lines have different insertion site effects, and you could imagine that these differences in patterns are actually being driven by completely different transcriptional profiles, such that in line A, you're using a whole bunch of transcription factors that are insensitive to the SNP, but in line B, you're actually recruiting DEF1 as part of your transcriptional complex, and so now we have the transcription factor that's actually sensitive to RS6295. So this is all a bit dissatisfying, I think. But it does essentially show us that RS6295 can indeed contribute to differences in receptor levels in vivo. All right, so what we know now from this entire story is that small changes in receptor levels affect behavior in mice, with an important sort of nod towards spatial and temporal specificity. That GG carriers have an increased risk for psychiatric hospitalization, that the G allele is associated with decreased transcription of HGR1A in the human prefrontal cortex, and that this SNP can indeed modulate receptor levels in vivo, although I think the jury's still out in terms of how exactly it does so and in what brain regions in humans. So, I've just shown you 2 stories that involve generating transgenic mice to investigate non-coding variants from other species. And I thought what I would do is kind of finish up with some of the lessons learned here. So I think we all know that mice are not tiny humans. So I'll start with the good. So the good is that I think this strategy of dual floxed and flirted loci within the same transgene can be a really good one for ensuring that you have sort of the same genetic background, two variants. And so I think that has a lot of promise from a design standpoint. The sort of ugly is this, that we know that insertion site matters for expression patterns, that transcriptional milieu or recognition sequences can differ across species. So if you start digging in the literature, humanized mouse transgenes are often expressed at much, much lower levels than the endogenous transgene. And the most commonly invoked explanation for that is sort of a mismatch between transcription factors and their optimal binding sequences across species. And I'm just going to add one here that I didn't show you any data for. So as this was going on, we wanted to ask the question of whether there was a SNP we could introduce into the endogenous mouse genome that would recapitulate the effects, at least in cell culture, and the short answer is no. So it turns out in the mouse genome, there's 2 DEF1 binding sites, and there was no mutation or series of mutations that I could introduce that in cell culture fully recapitulated the effects of the G allele. So I just want to add, as I'm finishing up here, that the other thing that I've learned, and I hope has made an impression, is that science is a warts and all business. I've definitely shown you some warts in my data today, but what I've learned along the way is that it's about asking the right questions. And certainly, I wish that I had made monogamous mice when I engineered in some sequence from voles. That would have been a very splashy story, but that's not how it worked out, but I think that we've learned a lot from that. And so one of the reasons why I am so grateful for having received this award today is that I think it recognizes someone, myself, who has not had a perfect career. My data has not come out beautifully and perfectly, and I certainly suffer from the same imposter syndrome that many young PIs, and maybe older PIs, I don't know, we'll see if I get there, suffer from. And so it's very validating to know that at least the quality of the questions that I've been asking seem to be the right ones. Okay, so the last maybe 3 minutes or so, I just want to tell you a little bit about what my lab is working on now. So we finished up and published that story on the humanized mice in January, and since starting at the University of Colorado, I've gone back to my original lab, so you may remember these guys from the beginning of my talk. And so we're very, very interested in sort of how does bonding change the brain and the transcriptome. And so these guys are monogamous voles, and the way that I think about this is that bonding leads to these profound experience-dependent changes in behavior. So why is it that if you're a prurivole, you mate, and subsequently your social behavior is changed drastically, such that you only want to hang out with one other individual, and you basically fight off everybody else? But if you're a metovole, you go through the same mating experience, and essentially nothing happens with respect to your social behavior. So one approach that we've taken is sort of a multi-pronged approach, where the first of this is to use in vivo calcium imaging to look at what's going on in the brains of these animals when they're interacting with their partner versus a novel individual. And so what I'm going to show you is just a quick video here. This was collected 2 weeks after the animals were introduced to each other and cohabited, and we knew that they had formed partner preference data I'm not showing here today. So this is the same as the beginning, except a top-down view of a partner preference test. And the test animal here actually has a miniaturized microscope on top of its head. And then what you're seeing here is a visualization of the neuronal activity that's going on within this animal's brain. And this is just using a genetically encoded fluorescence, calcium fluorescence indicator. So when we started doing this, I initially was like, oh yeah, we're in the nucleus of come in, it's reward, motivation, it's just going to light up when they hang out with their partner. That's obviously not what's going on here. And so this has led us to the hypothesis that maybe there's something special about the encoding of a bond that would be represented in these social interactions in prairie voles that's not present within meadow and in particular, we're investigating the hypothesis that there's a subset of cells that come to encode the bond that are reliably active when these animals are interacting with their partner. So this is just examples. So here's a partner cell. So this is a cell that's most active when the test animal is near its partner. This is a cell that's most active when it's near the novel individual, and then this is a cell that doesn't seem to have any sort of bias. And then we can map this here, and so the hypothesis is that these pink cells stably encode the bond, and that what you would see if you looked in a non-monogamous or promiscuous vole species is that you wouldn't see a reliable cell population that's activated during partner interaction. And so the way that we can functionally test this hypothesis is by using activity-dependent labeling. So just very briefly, we can go in and we can label those cells that are partner cells. And so this is done using a viral method here. This is just a control showing you that we did indeed infect the brain. And then these red cells up here are the cells that were active upon the introduction of a new mating partner. This animal was just put back into its home cage, so a social setting where it already knew other same-sex foals, so you don't see that. And so what my graduate student is interested in asking is, what are the transcriptional changes that occur within these partner active neurons? And so to do this, she's developed a trap vector that allows her to express GFP-tracked ribosome selectively within the neurons that are partner active. She can then pull this down and then send this for sequencing and ask, what are the gene products that are changed selectively within this ensemble that we think is encoding the partner? So just finish up with a couple of big picture take-home points here. So non-coding variation, unsurprisingly for this group, is a key source of individual and species-level differences in gene expression and behavior. But I would argue we still have a long way to go in terms of understanding how this variation functions within the cellularly heterogeneous context of the brain and across development. And you know, I've shown you two examples today where common genetic variation can sort of act as fodder for natural selection to shape individual differences in behavior within a population, potentially driving speciation, and that it may also contribute to risk for psychiatric disorders. And the last final point that I want to make is that I think I am very privileged to have gotten my faculty position when I did, in part because we're at a point in time where these weird organisms are not nearly as genetically intractable as they used to be. So there have been some beautiful talks and posters here showing that we have tools like CRISPR, which seem to work in pretty much any organism that you try them in. We have genomes, and we have, in mammals at least, some tremendous virally mediated tools that are available. This work was carried out across my graduate career with Larry Young at Edinburgh University and then Renee Hen at Columbia, both of whom have been absolutely fantastic mentors. And I would just like to finish by acknowledging the people who are doing all of the work now in my lab, and mention that I am looking for a postdoc in a tech. So if you have anybody looking to either gain a bit of experience before grad school or looking for a postdoc position on any of those projects I mentioned at the end, please send them my way. So thank you.
Yeah, so that was a really nice talk. I'm wondering, when you're monkeying around with the levels of the 5-HT1A, how is that affecting the other receptors, right? Because then, and also, you're also going to be changing the levels potentially of high affinity and low affinity receptors for serotonin.
Yeah, so the only one that we rigorously examined is the serotonin 1A receptor. So modulating autoreceptor levels does not appear to change heteroreceptor levels, which is somewhat surprising, and vice versa, but we did not look at the other serotonin receptors. So it's absolutely possible, especially with these manipulations that are changing global serotonin output, that some of these other receptors may be sensitive to that feedback.
I'm wondering if you looked at gene modulation and stem modification near your CG.
So we didn't. That was aim 3. And I struggled with whether or not, sort of how much depth to dive into, in part because it was clear that these humanized mouse models were not going to be the foundation of my long-term laboratory. Since they didn't look very mouse-like and they didn't look very human-like, all the studies that I had planned with respect to looking at sort of pharmacogenetic interactions or gene-environment interactions. So we didn't dig very deep on mechanism. Yeah, I completely agree, I think. And it's not up there now, but like one of the most intriguing things for me is that the G allele actually abolishes a CPG site. So you have built in the possibility for differences in methylation at that site.
I have 2 questions. You tell me whether those are right questions or not. First one is, did you look at sex-specific differences? Because it sounds like...
Yeah.
So... Consider. Second question is, did you look at the timing of the impact of early stress on especially effects of serotonin-related, receptor-related changes?
first question is sex differences. So I'll break this into two parts. For the vasopressin receptor story, we focused exclusively on males, and that's because vasopressin itself has a testosterone response element, so you have much, much higher brain levels of vasopressin release in males than in females. And manipulation of vasopressinergic systems in females in voles has no effect. So it seems to be a male-specific aspect to pair bonding. In the serotonin 1A receptor, if you go and look at the paper in the supplement, we go into some of the sex-specific effects. So there were, in certain behavioral tests and in certain receptors in certain brain regions, there were some effects of sex. It was messy, which is why I wanted to put in the supplement. But it does exist. And in humans, there are sex differences in serotonin 1A receptor levels. Second question was, early life stress. I took this out. When we did the analysis of the association study, one of the reasons that we were interested in that population is because it is a highly traumatized population, which is terrible, but from an experimental standpoint, it provides us with a very rich data set where you have a high proportion of individuals who have experienced early life trauma. And we did find a gene by environment interaction where we saw that essentially GG individuals were more sensitive to the effects of early life trauma. So if you sort of do a scale of trauma, they would reach the maximal level of psychiatric hospitalization faster than individuals with a C allele. We did not. So I also, in the transgenic, so the first set of mice where we decreased autoreceptor levels, there we did see a gene-environment interaction. So in the paper, there's also a subset of mice that were that underwent restraint stress during the same window as receptor knockdown, and we saw sort of an enhanced phenotype in those mice. We did not look at this in the humanized mice for the same reasons that I already sort of mentioned with respect to Carla's question.
Are the bulls, well, I guess how genetically heterogeneous are the bulls? And so, and the follow-up to that is really, how much phenotypic diversity do you see in pair bonding? Like, is there some aspect of that that you could use to further study the individual differences?
Yeah, so absolutely. So the short answer is they're outbred. The much longer answer is that all of the colonies in the US, I think at this point, are a bit bottlenecked. Just because to actually maintain a truly outbred heterogeneous population, you need colony sizes that are gigantic. So instead, what we do as a workaround is we kind of swap around our colonies on a pretty regular basis. So every couple years, I trade voles with somebody. That said, there is enough genetic variation that we should be able to harness that and look at behavioral variation, how it corresponds with behavioral variation. One of the things that I've always found fascinating about prairie voles is that when we run these studies and we look at partner preference, reliably there's about 10% that do not form a preference. And so we don't know the exact reason for that. It could just be that they don't form bonds because genetically they don't have whatever expression patterns in the brain enable them to do that. It could be that we do all arranged marriages in the lab and it's possible we just gave them a partner they absolutely hated, right? So there are different explanations, but we know that in the wild there is tremendous variation in this behavior. And that variation can actually be accounted for by the same gene that I presented here, albeit in a different brain region and a different polymorphism.
Didn't Larry just map it with an F2, essentially, or just a cross?
For which trait?
For the behavior of pair bonding? I thought there were some mapping studies that have been done.
So Steve Phelps has a really nice paper in Science that came out a couple of years ago that look at males and fidelity in the field.
Maybe it didn't go anywhere. We just heard it.
So Larry had a postdoc when I was a graduate student who was trying selective breeding for these guys to make ultra bonders and non-bonders. As far as I know, that and the follow-up heritability studies haven't been published yet.
Maybe I missed. So the receptor expression in the ball, the prairie versus meadow. So you think it's those promote insertion elements?
Say that again, sorry.
Those insertion elements in the promoter, right? Do you still think it's responsible for those differences in the expression level?
So the question was whether or not I think that the repeat element is responsible for the differences in receptor expression. So I'm going to answer this at two levels. So the short answer is yes, but only in specific brain regions. And the second is that I do think that there are some significant limitations to trying to model this in mice. And so I think that the appropriate experiment, one that's now feasible, would be to use CRISPR to delete that repeat-containing element in prairie voles and ask what happens to brain expression patterns. But I don't think that it's responsible for some of these key differences in expression, like the ventral pallidum, the retrosplenial cortex, etc. So I think it plays a role, but it's pretty minor in the grand scheme of things, and that it seems to be modulating behaviors I didn't talk about in depth, like stress coping and whatnot.
So can you tell us what that repeat is? I don't know if it's a microsatellite
yeah, so-
or a big bunch of transposons.
It's a series of microsatellites. So it's a series of di and tetranucleotide repeats interspersed with non-repetitive DNA. And so we did an analysis to look at what are the most common differences, because you can actually generate differences in length multiple different ways, and then try to use that to get a sense of which of those sequences might actually be important. There was at least one SNP that could explain the differences in the dentate gyrus, and then there was also variation in repeat length, where I think it was a GA binding factor. The actual sequence is in the paper if you want to take a look.
What was the range, you probably mentioned, what was the range of sizes there from smallest to largest?
Oh, it differs by about 30 base pairs, so not huge.
Are there any tricks you can use to get the metavole and prairievole to breed? And if so, could you then use that to find transacting factors that are
yeah.
responsible for the differences in expression?
And we tried some Barry Manilow. We did actually try this, right? Like the power of that approach is obvious. But the problem comes up in that they won't breed with each other, so we could try something like in vitro fertilization. But we know that they have different chromosome numbers, so even if we could get IVF to work, I would be very concerned that the F1 generation would be sterile. And so I think that's a dead end experimentally, unfortunately. That's why I'm always very jealous of the Parameskius people.
All right, thank you very much. Oh, sorry, never mind. We still have time for some questions. Go ahead, one more question?
I can catch it after.
Okay, all right, thanks.
So the executive committee is going to be meeting over lunch.